This is Bio 181, Chapter 17, and it's on biotechnology. So we'll start off with a few different concepts. The first one is on restriction enzymes. So these are enzymes. Here's our friendly Pac-Man. Remember, enzymes are generally proteins. And these enzymes are going to cut DNA. So DNA is our double helix, right? So double-stranded molecule made up of A's and C's and T's and G's and A's, T's and C's and G's and C's and A's and T's, right? So this enzyme is going to look for a specific sequence. So say it's going to look for A, C, T, G, A. Of course, that's just half of the DNA molecule. Say this is the 3' to 5' end. Then the other side of it would be the 5' to 3' end, and that would be our T, G, A, C, and T. So generally the restriction enzyme, it's an enzyme that has evolved often in viruses, in bacteria, and it evolved and it looks for sequences of viral DNA or viral RNA and chops up the genetic code of those viruses. So it's going to look for a sequence. It's going to cut down here. It usually goes across a little bit down the reading frame, and then it cuts here. So that will happen anytime it reads this specific sequence. So if you've got a long sequence of DNA and that sequence occurs here and here and here, then you're going to get 3 cuts. And you would get 1, 2, 3, 4 DNA fragments. They're different lengths. So say this is like 100 base pairs. This one might be 1,500 base pairs, BPs. This one might be 2,200 base pairs, and maybe this one's like 100 or maybe we'll just do 75. Okay, so why do we care about this? Well, when we get these fragments, then we can put them into a gel that is going to separate them out based on size. So if we put them all of these 4 fragments, 1, 2, 3, 4, into the top of this gel, and then put it in an electric field, since the DNA is negatively charged, it's going to move toward the positive charge. And the small fragment, this 75 tiny little piece, is going to go way down here at the bottom, whereas the heavier piece, this one that's 220 base pairs, is going to be slow and stuck up here toward the top. So by looking at a characteristic banding pattern, you might, have a crime scene, and you're going to say, what is the fragment pattern of the individual that did the crime that was found there? And then you'll have a series of suspects, and you're going to be looking for a pattern that matches up with that individual. So restriction enzymes have many functions, but that's just one of them. Polymerase chain reaction. So this is abbreviated, this is a biotechnology technique. It's abbreviated PCR. You've probably heard of a PCR test for COVID. It amplifies genetic material. So if you take a small sequence of DNA, what will happen in PCR is it's going to heat it up. It's going to separate it. That DNA will then be used, each of the DNA strands will be used to make a complementary strand. And then the same thing will happen again. So it cools down a little bit, makes the DNA, then it heats it up, splits it apart again. So then we've got here, what's going on? Yeah, so then we're going to do, if I stay with my color scheme, I guess, made it hard on myself. That's the bottom one. And then here, blue, splitting this into 2. There's blue on the bottom, my red on the top. Okay. And then we're going to cool it down. So each of these strands is then going to be used to make a complementary strand. So here now we've got some more strands. So we start off with one. After the first cycle, we've got two. After the next cycle, we've got four. So this increases exponentially. So very, very quickly, you're going to have, millions or billions of copies. Okay. So that is often referred to as amplification. So it amplifies DNA. That is a term that just means making a whole bunch of copies of your DNA. What's kind of cool about this is that the enzymes that work in very high temperatures. So the enzymes, the polymerase, remember polymerase is the enzyme that's going to come along and read the base pairs. Maybe they're going to be on this side, right? And then it's going to find the complementary base pairs that are around in the solution. Ooh, I'll show them in red here, and it's going to find them as it moves along. This enzyme can withstand very hot temperatures because it was isolated from boiling hot springs in Yellowstone. So it's a great application, an enzyme that is a DNA polymerase that can be heated up. When DNA is heated up, I should mention that it's the hydrogen bonds that are linking these strands that are broken in heat. So those hydrogen bonds linking the complementary bases are split apart, and then those are used to create a template for the new strand. All right, so next, gel electrophoresis. I really talked about this in the first example, so I'm going to go back. Gel electrophoresis separates DNA using an electric field. I'm just going to go back to the first slide here. When we made the different size fragments of DNA, we had the 100 base pairs, the 1500 base pairs, 200 or 2275. So we put that into a gel, and it's in an electric field. So there's a positive wire that's going to be running on the end of this gel here. There's a negative wire over here on this end. It's submerged in a buffer solution that conducts electricity. And so the DNA will be put into one of the wells. Here's another sample. It's put in here. And then again, it's going to be pulled down. Since DNA is negatively charged, it's going to be pulled down toward the positive end of the gel. The gel itself is kind of like a jello. It's a solution generally made out of a carbohydrate derived from seaweed. So it's just like a powder. You heat it up, it solidifies, put it in a form, and the DNA fragments will be separated based on size. The last thing for this video, I think it's the last thing, yep, is something called STRs. These are short tandem repeats. So these are found in all humans, and they're short sections of our DNA that don't actually code for any protein, and they're repeated over and over. And the number of times it's repeated is associated with, it's unique to each individual. So this individual here, you've got the sequence AGAT. So that's the short tandem repeat. So it's that same sequence. And for this one, individual A has it recurring time after time again. It's got 7 exact copies in a row. The second individual has 4, and then this individual here down here has 12. So there's lots of different forms of these. They can be in different places in the code, different places on the chromosome. There are certain sites where they're more common, and by targeting those specific ones, using restriction enzymes to actually cut these up, and then these are going to be of different lengths based on the individual. You're going to have banding patterns that are going to be specific to each individual, right? Depending upon how many of those sequences they have. If you've got 19 and say your restriction enzyme comes and cuts for something, there you're going to have a lot more fragments than if you only have 6. Okay. Oops, and that's all for this video. Got a little bit ahead.